Good morning, delegates from University of Sussex and colleagues from Mahidol. Today, I'm very happy to have a chance to talk to you about the new roles of the human papillomavirus protein that we have found in our lab. So, human papillomavirus is the cause of cervical cancer, as you might know. And among the 200 types of the HPV, around 200 types identified so far, type 16 is the most prevalent one found in women worldwide and also including Thai women. So in this study, we will focus on HPV 16. HPV 16 is a tiny DNA virus of only 8 kb, and when integrated into the host chromosome, they produce only two kinds of major proteins called E6 and E7. E6 and E7. Okay? But for type 16, it is a special type of human papillomavirus because the E6 protein can undergo splicing process to generate the two small E6 proteins called E6*1 and E6*2. Okay. Now, so when you normally call it E6, they will refer to both the full-length form and the truncated forms altogether. Okay, for E6 of 16 type. Now, what interests us is the report from Cosell in 2007, who said that E6 protein, the star one, has been increased when the cancer developed into the more advanced stage. So we are now very interested in this small protein and like to see how these small proteins involve in cervical cancer. And E6*1 is actually the major splice protein, comparing between the two forms, star1 and star2. Star1 is a major product. So that we start our work by doing the microarray. We're looking at the change of the expression level in the human cells after transfection with the E6*1 protein. Okay, E6*1 from HPV 16. And what we found is this list of genes, about 10 of them that were shown to be upregulated, increased in these E6* containing cells. So you will see that the first top two, and the last one actually are the enzyme coding genes. So we are more interested in these genes because we expect that it might be easier for us to follow the phenotypic change because they are enzymes. It might be easier for us to analyze. So that we start out our work by looking at the first two enzymes first. Briefly, the AKR enzyme actually is a group of aldoketoreductase. It's a big group of about 151 members. And it is responsible for the conversion of the carbonyl containing substrates into the alcohol. And in human, there are 4 subfamilies. Subfamily A, B, C, and D. For C, it refers to hydroxysteroid dehydrogenase as another name. So the AKRC type that are found to be increased in the E6* containing cells, again are divided into C1, C2, and C3. Depending on type of the substrates they prefer. And most of the substrate for the C type actually are the steroid hormones. So AKR1C then play important roles in the hormone action. This is C1 and this is C3. Now, to confirm, before we move ahead, we have to confirm that AKRC1 is the real target of the E6 star protein. So we confirmed this result again by using the real-time PCR, and we found that C1 protein is increased by the virus protein. This experiment has been done in the stable cell lines that we developed. These cell lines will express different types of oncogenes from HPV16, namely the short E6 star protein, or the full-length E6 protein, or E7, or combination of E6 star and E7, or combination of the three forms altogether, and another control from a different type of HPV. So you will see that E6 star 1 can upregulate the AKRC1 either as a single form or combination with E7. If you look at C3, they can also increase C3 level, but in much less induction ability. But no activity is shown for C2 at all. So when we look at the protein, we check for the protein, and you see that using the specific antibody to AKR1C, then you see again that cells expressing the E6 star protein significantly increase the protein product of the AKRC. Also, you can see some expression with the E7 and also some with the combination of the virus protein. So to confirm that the increase of this enzyme is really due to the virus protein, we then did the knockout experiment using the E6 shRNA. Then when we check the level of the E6 protein after transfection with the siRNA or shRNA specific for E6, then the E6 expression is dropped by half. And if you look at the enzyme level, C1 and C3 also drop, but not C2. So these results confirm that E6 is actually correlated with the C1 and C3 upregulation. So we'd like to know which residue in this virus protein that is responsible for this activity. So then we did the mutagenesis for 8 positions. The first group, for position 8, 10, and 11, mutation sites, these sites are based on the report for the full-length E6, where they found that these sites actually are responsible for nuclear localization signal required for the protein to enter into the nucleus, so then they can perform transcriptional upregulating activities. But the rest, the other positions, we based on the homology between the conserved sequence among the HPV types, so that we randomly pick up them. And as you can see from the RT-PCR result, that position 8 and 10 and 17 affect the activity of this E6 star protein because the production of AKRC1 drops. Now, number 10, 8, 10, and 17 are quite of our interest. So, one might ask the question if this mutation affects the nuclear entry of the protein. So we then check the localization of this E6 star variant. And as you can see that comparing with the wild type E6 star, all the mutant variants show the same pattern of localization. I mean, the same distribution pattern, which means that this is the fluorescent, and this is the nuclear staining, and this is a merge of the two. So that you can see that the protein spread all over. You can find in both cytoplasm and in the nucleus. So that the mutation in these three sites did not affect the localization activity of the protein. It is not so surprising because this protein is quite small, of only about 43 amino acids, so it may diffuse automatically through the nuclear membrane in and out. So to make sure that the protein that we study, actually we made a fusion protein with the GFP. We want to know if this fusion protein are still intact, and the signal that we follow is not just come from the control GFP alone, because GFP alone can also give the same type of signal, spreading all over the cells. So we did the Western blot or immunoblot to check whether the proteins are still intact, and we see that all the proteins are still the fusion protein. Now we look for the activity of this HPV protein on the C1 promoter. We made a construct, deletion construct, and you will see that from 500 upstream region of the C1 promoter, you can see that the E6 star wild type protein can show about twofold upregulation activity over the control. But when they have mutations at position 10 and 17, the activity of the luciferase reporter genes drop. So again, it confirms our previous result. So when we delete out the AP1 sites, and the activity was not affected much, meaning that these 4 AP1 sites may not be important for HPV-mediated transactivation activity. But when we make mutation on the SP1, you will see that this is the basal level of the cell. The basal level of the cell also dropped by half, and also in the presence of the virus, the activities also drop. Meaning that SP1 site is important for both the basal level of the AKR1C promoter, the activity, and also for the virus to mediate their action. So at this stage, we can conclude that SP1 site on the AKRC1 promoter is partially involved in this HPV16 E6 star mediated transcriptional upregulation activity. And probably the rest can be due to these factors. So we asked if the increase of the AKR1 enzyme results in any phenotypic change, then we test for the drug sensitivity, because the enzyme is found to be increased in many of the drug-resistant cancer. Okay, so that we then test for the sensitivity to 2 different kinds of drugs. One is doxorubicin, and another one you heard before, etoposide. These 2 drugs are actually in the same group of topo-2, topoisomerase 2 inhibitors. So by using MTT and colony-forming assays, we show that in cells containing E6 star protein, the drug resistance occur because you need twice the number of viable cells are found than the control vector. So meaning that they're quite resistant to the drug. But when you have introduced the mutation, for example, mutation on residue 8, 10, and 17, as we shown before, the sensitivity, the number of the viable cells drop to comparable to the control, meaning that they do not confer any drug-resistant property when they mutate, when these positions are mutated. And both assays confirm each other. Okay, so when we look at the protein in this mutant, again we see that the enzymes drop as well in this mutant. So for the conclusion of this part, we can say that the new role of E6 protein in enhancing the expression of the enzyme aldoketoreductase is found. And these elevated level of AKR1C proteins are sufficient to confer resistance to a certain type of anti-cancer drugs. And position 8, 10, and 17 are the key residues necessary for this activity. And lastly, E6 can mediate transactivation activity partly through the SP1 binding sites on the AKRC1 promoter. Now, if you look at this diagram, Tang Chi Kun, 2008, has summarized a variety of E6 target proteins. As you can see that the E6 is a very special virus protein that it can attack many of the cellular proteins, including in a variety of mechanisms. For example, proteins involved in the transcription, proliferation, cell-cell adhesion, differentiation, immunity, ubiquitination, and apoptosis. The E6 from the HPV16 can interfere with all these cellular targets. And now we add up one more, that the target of the E6 in the form of E6 has been found, that is the AKR1C. And this will bring the phenotypic change for the drug-resistant behavior. Next, we move to the last enzyme here, the MMP10. So MMP, again, is a big group of enzyme. It's a zinc protease, which contain about 23 members in human. And this MMP can degrade most of the extracellular matrix in human cells, and this degradation will promote cell invasion and metastasis of the cancer. Okay. Now, we like to start out the second part by just looking first at the invasion property of the cervical cancer cells containing HPV versus the cervical cancer cell without the HPV. Now, so the first two cell lines are HPV negative. 293 is not the cervical one, but it is used as a control of the HPV negative too. The last three cell lines are HPV positive. The first two contain 16 and the last one contain 18 type. So that we look for the expression level of the MMP. We select 7 type of MMPs to look in our study. These 7 MMPs were reported to be increased in cervical cancer. So then we select to look for these 7 MMP, namely MMP1, MMP2, 7, 9, 10, 11, and MT1. And you can see that MMP2, 7, and MT1 increase in the HPV positive cells. So then we concentrate on these 3 MMPs. Now, again, we use the stable cell line expressing different types of the HPV oncoproteins to look for the correlation, the relationship between HPV oncoproteins and the expression of these 3 MMP. And you can see that type 16, no matter what they are present as a single form or combination form, all forms of the HPV oncoproteins can upregulate MMP number 2 and MT1, but not number 7, which confirm the previous result. Oh, no, not yet, sorry. So this one just tell us that only number 2 and MT1 that are correlated well with the HPV oncoproteins. Whereas oncoprotein from other HPV type did not do this job. So this is quite specific for type 16. Detection of the HPV expression by RT-PCR and also activity of the HPV oncoproteins in p53 degradation were also tested to confirm that the virus protein is really expressed in the cell of our study. Next, we like to see if the invasion ability is related to the increase of MMP2 and MT1 enzyme or not, because this is the enzyme responsible for cell invasion mostly. Then you will see that cell expressing different type of virus protein also show high invading cells when we test for invasion assay using in vitro invasion test, and also by wound healing as a confirm, we get the same results that all the virus protein seems to show the invasiveness. And the pink arrows show you that the combination of the virus protein E6, 2 forms of the E6, full length and the short form. And E6, E7 contain all 3 forms, okay? And E6 and E7 only 2 forms. These 3 combinations always show a higher number of invading cells or higher invasiveness in both assays. So we move on to detect the expression of the enzymes in these cells. MMP2 is detected by using the seismography because it is a gelatinase and it is easily detected using enzyme gelatinolytic activity. As you can see that they also confirm that the virus oncoproteins in the combination form show higher MMP2 activity. When you look at MT1 expression by Western blot, again, the combination form of the virus show better expression of this enzyme. Now, again, we want to make sure that the MMP is responsible, E6 star is responsible for upregulating the enzyme. So we use the knockdown experiment again. We use the Shiha cell. Shiha cell is the HPV16 containing cell line. And we knocked out the E6 protein using the shRNA technology. First, you see that we check the level of the oncogene that has been expressed in this knockdown cell line, and you will see that all E6 full length, E6 star, and E7 levels are dropped. Same as when we check for the MMP activity in this knockdown cell line, only MMP2 and MT1 show the effect, but not MMP7. Again, we test for the activity of this knockdown cell line using the seismography to look for the MMP2 activity. You can see that the knockdown cell line has decreased MMP2 activity, same as decrease of the MT level detected by the immunoblood. And also, these correlate well with the invasion test when we look at the invasion ability of these cell lines after knocking down with the shRNA of the E6, the invasion ability drops. So now we are quite certain, we're quite sure that E6 star and E6 full length and also E7 in combination form, they play important role in upregulating these two enzymes and also responsible for cell invasion. One might ask the question if we are really sure that invasion of the cancer cells that we see are actually due to MMP2 and MT1 or not, because there are so many MMPs that are produced in the cancer cells. So we use the inhibition assay using the specific inhibitor for MMP2 and specific inhibitor for MT1, that is the antibody actually. The results confirm that in the presence of MMP2 inhibitors, the cell invasion drops in a dose-dependent manner, both in the stable cell line expressing E6, E7 protein or in the natural Shiha cell line. Same result was obtained when we used the antibody specific for MT1. So at this stage, we can say that the invasion that we found in these cell lines are due to the activity of MMP2 and MT1. Okay? Look at this molecule TIM2. MMP2 requires activation by MT1. These two enzymes have to work together. But in order for MMP2 to be activated by MT1, they need a linking molecule or the bridging molecule called TIM2. So TIM2 will bring the two enzymes together to form the ternary complex. And this complex then activates the MMP2 to be active protease. So that we then look for the expression of the TIM2 to see whether this linker, the expression of the linker is also increased in these cells. And the results show that, yes, TIM2 is also slightly increased in all HPV protein expressing cells, both at the protein levels and the RNA level. So that it's not so surprising to see this because some of the cell lines also show elevated level of TIM2 in invasive stage of cervical cancer cell too. And we surmise that the increase of the TIM2 might also provide a proper ratio between MMP2 and the MT1 in order to make a better activation of MMP2 enzymes. But whether the HPV oncoproteins also upregulate this gene or not remains to be studied. We don't know at the moment. Is it the right direct effect or not? Promoter analysis was also performed using similar strategy, a deletion constructs, and you can see that HPV oncoproteins, and this time we use a combination form E6, E7. They behave the same way on both promoters. As you can see that the long constructs of about 1,200 kb in MT1 promoters, they can upregulate gene expression. They can upregulate the luciferase reporter gene. But when you delete it out, the first group of the transcription factors, you see no effect. But when you delete the second group containing PIR3, then you see a dramatically drop of the activity, meaning that PIR3 here should be responsible for the virus activity. Same as in MMP2, with the long construct of about 1,700 kb, you see more than 2 times of activation. But when you delete out the extremity binding sites here, you see very small effect. But when you remove the double side of the PIR3, and then you see a significant effect. So this result show you that PIR3 in both MMP2 and MT1 might be the site of action of the virus protein on these two promoters. And further analysis of the promoter of these two promoters are ongoing in our lab. So lastly, we like to see if the phenomenon that we see in vitro study would be also the case in vivo. So we are lucky that we are provided samples from cervical cancer patients are kindly provided from Ms. Jaranya Ngamkham from the National Cancer Institute Thailand, who is sitting somewhere here. So she then provided us a sample from cervical cancer patients to follow the expression of MMP2 and MT1 enzyme. And as you can see here that the red or the violet one, the violet color represents MMP2 and MT1 enzyme, whereas the blue ones represent the nuclei staining. This is a phase contrast. So wherever there is MMP2 or MMT1, there is expression of nearly all of cells express this enzyme. So when we summarize the data in about 30 samples that we obtained, these samples, half of them contain HPV-16 and another half contain other types of HPV or HPV negative. You will see that in HPV positive cells, the expression of MMP2 was quite high, as 87% was detected, expression of MMP2, whereas 93% for MT1 was detected. The samples was kind of based on HPV infection without, regardless of the pathological staging. We don't care for the staging. But if we consider only the invasive samples, we remove some that are not invasive stage out. Yes, only the invasive ones are counted. The number are now changed from 87 to 92% and from 93 to 100%. So this number increased even more when you only correlate with the invasive stage. So from this data, we conclude that the invasiveness of the cervical cancer is correlated well with the expression of the MMP2 and MT1 protein, and these might be due to the HPV oncoproteins produced by the 16 type. So in conclusion, what we found in the second part is that HPV oncoproteins can also upregulate transcription of the MMP genes. MMP2 and MT1 are examples, and combination of the proteins show better effect. And the result is confirmed in vivo. Also, the binding sites for PL3 and SP1 seems to be essential for this activity. And lastly, one more activity of HPV1 core protein is added here, that is the invasion activity mediated by MP2 and MT1. So in summary, we like to say that actually human papillomavirus protein can do more than we think in promoting cervical cancer. And as we shown here, they can promote the aggressiveness of the cancer. And last but not least, I'd like to thank for all the support from my students, and particularly the 4 people here who mainly contribute in this work. And also the financial support. I really appreciate the support from Biotech, NASDA, and Faculty of Science, Mahidol University, and also the NRCT. Thank you very much.